morning, students. Welcome back to our official YouTube channel, SR and BGNR Government Degree College, Khammam. Myself, Dr. B. Satish Kumar, Assistant Professor in Biochemistry. Today, we are going to continue the last video lesson, that is replication, DNA replication in prokaryotes. In the previous video lesson, we have learned about some basics of DNA replication. Introduction to DNA replication. In that, the first topic we have covered so far is uh, the experimental evidences to prove nucleic acids as genetic material. Okay, so in the last video lesson, we have covered some basics about the structure of double helical DNA structure. In that double helical DNA structure, let us have some quick review about quickly recall some basics of DNA structure. If you understand the DNA structure, then only we can understand the replication process. In DNA structure, we will have two strands running in opposite direction. They are two anti-parallel strands. Each strand contains nucleotides, deoxyribonucleotides as monomeric units, and they will be joined to each other by means of phosphodiester bonds to form a single strand. One strand interacts with another strand by means of hydrogen bonds. Here in nitrogen, we have two different kinds of nitrogenous bases, purines and pyrimidines. Purines are adenine and guanine, pyrimidines are thymine and cytosine. So here the purines always interact with the pyrimidines because purine is having two-membered ring structure. Pyrimidines are having only one-membered ring structure. So therefore the length of the size of purines is more than the pyrimidines. To have a uniform structure throughout the length of uniform width throughout the length of DNA molecule, the purines must interact with pyrimidines. Hence, here the adenine interacts with thymine by means of two hydrogen bonds, and guanine interacts with cytosine by means of three hydrogen bonds. This is the basics of double helical DNA structure. So that is about the basic structure of DNA. Double helical DNA. DNA acts as genetic material, as we all know about it. The first topic in DNA replication unit is the evidences, the experimental evidences which proved DNA as genetic material or nucleic acids or genetic material. So we have two experiments conducted by different investigators which proved the nucleic acids or acts as genetic materials. The role of the nucleic acids is genetic material. We have four different types of biomolecules in each and every living cell. Number one, carbohydrates, then lipids, proteins, and nucleic acids. So each of these biomolecules have different roles in their cells. But in case of nucleic acids, the nucleic acids acts as genetic material. Genetic material means that carries the genetic information. That genetic information is nothing but it contains genes which encodes proteins, and those proteins are very essential for the existence and maintenance of the life. If we take any example of any metabolic pathway like glycolysis. Glycolysis depends on 10 different enzymes. So if the cell is having 10 those 10 essential enzymes for the glycolysis, then only the glycolysis, the glucose which entered the cell enters into glycolysis, and ultimately it forms pyruvate from the glucose. So the information for the synthesis of these enzymes, enzymes are nothing but proteins. So therefore, the synthesis of these 10 different proteins requires some information which contains the information which is essential to have the amino acid sequence in the specific proteins, because all proteins are different from each other only by means of primary structure. So because of the primary structure, the secondary structure, tertiary structure, quaternary structure, and the function of the protein also changes. So primarily it depends on the sequence of amino acids. So the sequence of amino acids, the information for the correct sequence of amino acids in each and every protein is present in the form of genes. So one gene produces one polypeptide chain. That polypeptide chain may become an enzyme or a subunit of an enzyme. So therefore, without genes, no information will be there. Without any information, no proteins are synthesized, and no metabolic processes, and life cannot exist or maintain itself. So that is why the information is very, very essential thing for the life. Therefore, the information is kept in the form of nucleotide sequences in DNA. So that was proved by two different experiments conducted by different investigators. One of those experiments is Griffith's bacterial transformation experiment. We have already discussed about the experiment, how the experiment was conducted, and what are the outcomes of experiment. So in short, we can say that in the bacterial transformation experiment, the rough strain, which is a virulent strain, is converted to virulent strain by transfer of the genes responsible for the virulence in smooth strain. So here, when we injected heat killed, when we mixed heat killed smooth strain, so that means that bacteria is killed, and but the DNA or the genetic material, whatever it may be, whether it is a carbohydrate or lipid or protein or nucleic acid, whatever it may be, the information is still there, but the cells are dead. Now these cells, dead cells, with their information molecules are mixed with live avirulent non-pathogenic rough strain. So what happened here is the genetic information in the form of some molecule is simply transferred into rough strain. When we mixed both these solutions, here the genetic material entered into the rough strain, then rough strain becomes smooth strain, because now the rough strain is having the information for the virulence. The virulence factor, in fact, is that can be attributed to the presence of lipopolysaccharide layer on the cell membrane of bacterial cell. So the synthesis of lipopolysaccharide layer is governed by some of the genes, and those genes are directly transferred to live avirulent rough strain. So now the rough strain becomes smooth strain. Because of that, that smooth strain is able to kill the mice, kill that mouse which was infected with the solution. So here the transformation of rough strain to smooth strain taken place only by the transfer of genetic information from smooth strain to rough strain. So this is called Griffith bacterial transformation experiment. After that, three other investigators named Avery, McLeod and McCarthy, those three investigators further investigated into this same experiment, and they found out that the transforming principle, the transforming genetic principle is nucleic acid. Nucleic acids are the genetic material. So they uh, proved with evidences. After that, we have one more experiment that visualized the presence of nucleic acids are very essential for the life cycle of bacteriophages. So in one set, the scientists used bacteriophage, which is the bacteriophage will have a outer protein coat, and inside the protein coat capsid it will have nucleic acids, the genetic material. So in an attempt, they wanted to find out which one is essential for the life cycle of virus, whether the protein coat is essential molecule or the nucleic acids, which one is the genetic material for the life cycle or production of several virions from the bacteriophages. So in one experiment, they used protein coats which are radio labeled protein coats of bacteriophage, and in another set of experiment they used radio labeled nucleic acids. When these bacteriophages are allowed to infect their host cell, E. coli cell, and after some time they have taken those solutions and blended it, and therefore they removed the the bacteriophage proteins from the bacteria. So the bacteriophage comes and attaches to the bacteriophage cell wall, cell membrane, and injects the nucleic acids into host cell. So when they blended the total solution, the attached protein coats will be removed, easily gets dissociated from the surface of E. coli cells, and after that solution was subjected to centrifugation. In centrifugation, we can easily separate the protein coats of bacteriophage and the E. coli cells into two different bands. So in this set, these bacteriophage protein coats are were separated. So now we have only cells. When we analyze these cell population under autoradiography, they found the radioactivity in the protein coats only. That protein coats are really separated. So there is no radioactivity inside the cell. But in the second set of experiment, they had radioactivity inside the cell because the nucleic acids entered into the cell. So now they allowed these two batches further for the commencement of lytic cycle. So after some time, the bacteriophages are produced from the cell and they released by breaking the E. coli cell membranes. So that means here in the first set, the protein coats are removed, but still these cells are producing virions, functional virions. Here also, here the protein coats are not there, but the nucleic acid is there inside the cell. It is producing virions. So that means the nucleic acids are essentially genetic material, acting as genetic material for the production of virions. It is not the protein coat that acts as genetic material. So this is how they experimentally proved the nucleic acids as a genetic material. And one more experiment you have learned in the previous class is Michelson and Stahl experiment, and this experiment is used to prove that DNA replication is semi-conservative model. Semi-conservative means that DNA, the parent DNA will have two strands. The parent DNA is having two strands. These two strands will be separated. So one strand here, another strand here. Two strands will be separated during the replication process, and the DNA polymerase is an enzyme which is essential for the synthesis of new DNA molecule on the old existing DNA. So now this single strand acts as template. On the template, a new strand will be synthesized, making it a double strand. So here also a new strand will be synthesized. So that means semi-conservative model means each old strand acts as template around which a new strand will be synthesized. So old strand is guiding the synthesis of new strand. So in the daughter cells, in the two daughter cells formed from the parent cell, will have one old and one new strand. So new strand will be synthesized by following Watson-Crick base pairing rules. A interacts with T, base pairs with T by means of 2 hydrogen bonds, and G base pairs with C by means of 3 hydrogen bonds. This is known as semi-conservative mode of replication. And when we come to the actual process of replication, we conveniently divide the replication process in E. coli cells into three different stages. First stage is initiation, second stage is elongation, and third stage is termination. In the stage of initiation, the DNA replication process will be initiated at a particular site. So it is not a random process. The initiation, the DNA replication doesn't start at a random site. There is a specific site on each and every DNA molecule of E. coli cell. So E. coli cell is having one circular DNA molecule, and that circular DNA molecule is having one initiation site. At that site only, all the replication machinery, the proteins which are involved in the replication process, comes and binds there, and they initiate the DNA replication process. So that is called origin, the region of origin. That sequence is uh, named as ORI-C sequence. So initiation begins at origin sequence, and all the uh, protein machinery necessary for the DNA replication process will come and assemble there. And the second stage is elongation. So once the initiation of replication takes place, now the DNA polymerase gradually moves along the DNA strands, separated old DNA strands, and synthesize new DNA strand around each old strand. The synthesis always takes place in 5' to 3' direction, utilizing the template strands in 3' to 5' direction. The elongation is also again subdivided into leading strand synthesis and lagging strand synthesis that we will discuss elaborately later. Here, leading strand and lagging strand. So one strand is called as leading strand, where the new strand will be synthesized continuously, and in case of lagging strand, the other strand is known as lagging strand, and the new DNA molecule is not synthesized in continuous fashion, it is synthesized discontinuously. So that means the short DNA molecules will be synthesized along the lagging strand. So those fragments, short DNA fragments, are known as Okazaki fragments, and that strand follows discontinuous method because of the orientation of the strand. And finally, we will see about the termination process. Like initiation, termination is also very essential, and there is also a specific point where the termination of replication takes place. Like the origin sequence, for the beginning of the replication process, we have termination sequence in the E. coli cell DNA for the termination of replication process. So everything at the termination site, every protein assembly will dissociate, and that marks the finishing of DNA replication process. So once termination takes place, two DNA molecules will be formed from one DNA parent DNA molecule. So these are three processes that we are going to discuss in detail. The very first one is the initiation of replication. So here, if we look at this diagram carefully, an E. coli cell contains a circular double-stranded DNA molecule. So this is, suppose, the circular DNA, double-stranded DNA. So at somewhere here, the origin sequence is there. This is origin sequence. So this origin sequence will have specific sequences. One specific sequence, that specific sequence or the part of origin sequence. Those sequences are known as consensus sequences. So at this site, what happens, that the proteins which are essential for the DNA replication comes and binds here and initiate the replication process. So before going to discuss elaborately about the initiation process, we have to know, we must have some knowledge about some of the proteins which involve in the replication process, initiation of replication process. So those proteins like so here these are main protein important proteins involved in the replication process. So first one is helicase. This is helicase protein. A helicase is a protein that unwind parental double helix at replication fork. Replication fork means when it opens, when the uh, when the circular double stranded DNA. So this is the circular double stranded DNA. So what happens, suppose we have origin sequence is here, then here the opening of, so here at the site, at the origin site, a bubble is being formed. So this kind of bubble is formed. So that means now the two strands will be separated. So it looks like theta symbol in mathematics, and this the the junctions here, the junction here, yeah, the junction, these junctions are known as replication forks. So when we start unwinding, the double stranded DNA, so here this is the replication fork, and these two strands are being separated. These are bound base paired double stranded DNA. So these base pairing has been disrupted and two strands are separated. So now the separation needs a protein, and that protein is the helicase. So a helicase binds one of the old strand and moves along the DNA molecule. As it moves, it breaks the hydrogen bonds between the two strands. So now the two strands will get separated. So the activity of the helicase and function is separation of double strand. So once the strands are separated, they may come back and reassociate with each other easily because of complementary bases on the opposite strands. Okay. So here what happens to prevent the reassociation of these two separated strands, it requires another protein called single stranded binding protein. They are also known as SSB, single stranding binding protein. So these single stranding binding proteins binds to the separated single strands and stabilize them, and they prevent reassociation of the single strands. Okay, so these two are essential proteins in the initiation process of replication. Along with these two, the DNA polymerase is also an important enzyme which which is for the synthesis of new strand. And so one more enzyme is also there, that is a primase, that is primase enzyme. So this primase enzyme here, this is the primase enzyme. This is nothing but a RNA primase. Like DNA polymerase, the RNA primase synthesizes short nucleotide sequences of RNA, short RNA strands, RNA primers, RNA primers. So that means, suppose this is the separated strand. Now the DNA polymerase cannot initiate the synthesis of new strand on the old strand directly. It cannot start the synthesis, but instead it can elongate already existing molecules. So therefore this the RNA primase synthesize a RNA primer molecule, and these RNA primer molecules will be extended by DNA polymerase 3. So for the initiation of nucleotide synthesis, new strand synthesis, we require a RNA primase enzyme that will prime or that will synthesize short RNA primers, and these short RNA primers are extended by DNA polymerase 3 enzyme. Okay, so therefore in the initiation we require helicase for the unwinding of double stranded DNA, single stranded binding protein to stabilize the unwound single stranded DNA molecules. Third one is RNA primase to synthesize RNA primers which can only be extended by the DNA polymerase. Otherwise DNA polymerase cannot initiate the synthesis directly. It requires uh, something some molecule, already existing molecule, then only it binds to that molecule and just extend it but not initiate it. Okay, so another important protein is also known as topoisomerase. This topoisomerase actually, during the replication process, during the unwinding by the helicase, if the helicase unwinds the double helical DNA molecule, then what happens? Because of unwinding, a supercoil will be formed in front and back also, in front and back of the DNA molecules. So these supercoils presents some stress, topological stress, which prevents the further movement of helicase and as well as DNA polymerase. So therefore, this topological stress will be relieved by topoisomerases, and that relieves the supercoils, removes the supercoils, so that the helicase easily moves through the double helical DNA and unwinds it. Okay, so here we have helicase enzyme, single stranded binding protein, topoisomerases, and primases, and DNA polymerase 3, of course. These 5 different molecules or proteins and enzymes will be involved in the initiation of DNA replication. So here, this is the origin sequence. This is the origin sequence, ORIC. So this ORIC will have 2 regions. One region is having 9 base pair repeats. 9 base pair repeats. So that means here a 9 base pair sequence will be there, and that same sequence will be here, here, and here. So we have 4 regions, subregions, which are having 9 base pairs. Same sequence, 9 base pair sequence, and besides this, it is having 3 more sequences which are 13 base pair repeats, which are AT rich sequences. So these 2 regions are collectively known as the ORI sequence. The 9 base pair repeat is called these 2 regions are called the ORIC sequence. So ORIC sequence is the region where the DNA unwinding takes place, and initiation of replication also takes place. So here the 9 base pair repeats are bound by DNA A protein. Okay, here DNA A protein is there. These green color balls are DNA A protein. So the DNA A protein that recognizes this 9 base pair repeats, they come and bind to these 9 base pair repeats, and they interact with each other, so that the DNA will be wrapped around wrapped around the DNA A protein complex. So now, because of this wrapping, the AT-rich region will be at the junction, at just very closer to the wraparound. So because it is AT-rich, the base pairing between these two strands will be weak, because the A interacts with B by means of only two hydrogen bonds. The AT base pair is weaker than GC base pair. So that is why this region easily gets unwinding. So now this causes unwinding. This uh, unwinding, in other sense, it is also called as denaturation. The denaturation of double strand takes place at the 13 base pair repeat region. So now, as the unwinding takes place, what happens? The helicase comes and binds to this one of the strand, and that moves along the double strand. So as it moves, the double strand will be separated, unwound to form single stranded region. So here, this purple color ring structure, here we can find it. So that is, it is bound to this and moving in this direction, and here also there one helicase and another helicase. One helicase is bound to this strand and another helicase is bound to the opposite strand. This will move in this direction, this will move in this direction. So this junction is known as replication fork. So we have one replication fork and another replication fork. So now the DNA replication takes place in two directions, this direction and this direction. One helicase moves in this direction and all the essential proteins will be assembled here, and it moves in direction, and the same thing happens in this direction also. So that is why the DNA replication is bidirectional. Okay, so what happens here? Suppose this is the double stranded DNA, E. coli double stranded DNA. What happens? One replication machinery moves in this direction, another will move in this direction. So that means we have two replication forks, two replication assemblies, and bidirectional synthesis. Okay, so now here you can see these yellow color proteins. These yellow color proteins are nothing but single stranded binding proteins. So these single stranded binding proteins will bound to separated single strands in order to prevent the reassociation. If there are no single stranded proteins here, then what happens? These two strands once again they will interact with each other, and they will winds again, wound around each other and form double stranded structure. So to prevent that, we need single stranded binding proteins. Okay, so this is what the initiation process. So now, once replication forks, these bidirectional two replication forks are formed, the RNA primase attaches to the replication forks at both region, here and here. So this RNA primase synthesize a RNA primer here and another RNA primer here. So now the DNA polymerase binds here and binds here. It will extend this way, this will extend this way. Okay, so this is what the initiation. In the initiation, we have origin sequence and DNA A proteins which recognize the origin sequence. Then the helicase which unwind double stranded DNA, making it single strands, and the single strands will be stabilized by the single stranded binding proteins. So that is initiation, and the next one is elongation. So in the elongation of DNA replication, elongation is nothing but the synthesis of DNA new strand on the old strand. Old strand is now called as a template strand. A template strand is a guiding strand on which a new strand is formed that guides the nucleotide sequence on the new strand. So based on the nucleotide sequence, based on the base sequence in the template strand, the base sequence will be synthesized in the new strand. For A, the new strand will have T. For G, the new strand will have C. Okay, so this is what elongation. In the elongation, the DNA polymerase will elongate the RNA primers. We in the initiation we have seen that replication is not initiated. The synthesis of DNA is not initiated on its own by the DNA polymerase. It requires the assistance by RNA primase enzyme. RNA primase enzyme now synthesizes a RNA primer. So this is the primer. This is the template strand. So now the DNA polymerase, what it does, it will join a new nucleotide. It will add a new nucleotide at the 3' hydroxy end of the RNA primer. So here a phosphodiester bond is formed between the hydroxy group of lost nucleotide and the phosphate group, 5' phosphate group of the incoming nucleotide. Okay, so a new phosphodiester bond will be formed. So the substrate for DNA polymerase is DNTPs, deoxynucleotides triphosphates, deoxyribonucleotide triphosphates. So that means it is having 3 phosphates at the 5' end. So during the process, during the formation of phosphodiester bond, what happens? 2 phosphates in the form of PPI, inorganic pyrophosphate, is removed and only 1 phosphate now will be present in between the 2 nucleotides. So that is known as a phosphodiester bond. A phosphodiester bond is formed. So the substrates are DNTPs, and to form a phosphodiester bond, 2 ATPs are required. 1 PPI is equal to 1 PI equal to 1 ATP, and 2 PIs, that is PPI, is equal to 2 ATPs. So for each and every addition of 1 deoxynucleotide requires 2 ATP molecules. The energy of 2 ATP molecules. Okay. So and in the elongation, if we look at this image here, the diagram, so this is called the leading strand. This is called leading strand and this is called lagging strand here, because the leading strand so now it is running in 3' to 5' direction. Okay, so from here to here, 3' to 5' direction, 3' to 5' direction. So in the same way, the other strand, the new strand, that will be in anti-parallel direction, so that in opposite direction. So therefore it will be in 5' to 3' direction. So the DNA polymerase always adds nucleotides, deoxynucleotides, only in 5' to 3' direction. So the synthesis always takes place from 5 prime to 3 prime direction, copying or reading or utilizing template in 3 prime to 5 prime direction. Template will be in 3 prime to 5 prime direction. Newly synthesized strand will be in 5 prime to 3 prime direction. Okay, so this is the synthesis of leading strand. And when we look at the other strand, so there is a problem. If it runs in 3 prime to 5 prime direction this way, it runs in the same direction, it runs in 5 prime to 3 prime direction in other direction. So now the DNA polymerase cannot move along the strand in opposite direction. The DNA polymerase always moves in only one direction on the both template strands. So that is why it cannot synthesize the lagging strand. So for that it will loop the lagging strand, template, and because of that looping, only short stretches of DNA molecules will be synthesized. So each and every short stretch will contain a small RNA primer extended by DNA polymerase 2, 3 to form that short stretch of DNA, and another primer, another DNA stretch, another primer, another DNA stretch. So in that way we will have very short stretches of DNA, newly synthesized DNA molecule. So this is called discontinuous synthesis. So the lagging strand will be synthesized in discontinuous manner, but the leading strand is continuously being synthesized. The continuous synthesis takes place on the leading strand. So there is no stopping and lagging for some time and restarting the synthesis again. There is no such thing in case of leading strand, but in case of lagging strand, a stretch of DNA is synthesized, then again the DNA polymerase stops, the RNA primer comes and synthesize a small primer, and then again the DNA polymerase extend that newly formed RNA primer and once again stops, once again RNA primer comes. So this goes on in a cyclic manner. So therefore we always have the small sized fragments, DNA fragments, which are called as Okazaki fragments. Okazaki fragments first I discovered by Okazaki. So therefore they were named after his name, Okazaki fragments. Even though they are synthesized in fragments, the DNA polymerase 1, that is the repair enzyme that removes RNA primers and replace with DNA molecules. Okay, so then another enzyme is there. That enzyme will seal the gaps between these two Okazaki fragments. The gaps between the Okazaki fragments will be sealed by an important enzyme known as DNA ligase. So this is the DNA ligase. Okay, DNA ligase enzyme using ATP for energy, and this is the DNA strand 5 prime 3 prime hydroxy group. This is another DNA fragment. Suppose this is one Okazaki fragment, another Okazaki fragment. A phosphodiester bond is formed between this hydroxy and phosphate group. Okay, the gap is sealed by the enzyme called DNA ligase. So the Okazaki fragments are formed, and Okazaki fragments are the RNA primers are removed by DNA polymerase 1, and the Okazaki fragments are joined by DNA ligase. So this is called leading and lagging strand synthesis. Okay, so now last topic is last stage is termination. So once the DNA polymerase reaches a termination region, like that of origin sequence, it has a termination sequence which is known as a ter sequence. So it is having some sequences TER A, B, C, and D sequences. These TER sequences are recognized by TUS proteins, terminus utilization sequences substances, and these TUS proteins recognize the TER sequences and they will mark the termination of replication process. So once the replication fork reaches termination sequence, because of TER-TUS complex, all the protein assembly from the DNA will get dissociated and replication will be stopped. So that is the end of the replication. And now we will have two DNA, double helical DNA structures in synthesized in semi-conservative mode. Each DNA molecule will have one old and one new strand. And the final topic in DNA replication is the inhibitors of DNA replication. Most of the inhibitors, actually this topic is also important to study because the inhibitors are will act as antibiotics. So if we use these DNA replication inhibitors, they will inhibit the replication of bacteria, and that leads to the death of those bacteria. So we can use those inhibitors as drugs which kill bacteria. Those are known as antibacterial or um, antibacterial drugs. Okay, so these are some of the inhibitors, antibacterial drugs or antimicrobial drugs generally used to prevent infection caused by bacteria. These are like doromycin and adriamycin and actinomycin D, ethidium bromide and proflavin, novabiacin and oxolinic acid, afidacolin and rifampicin. So these are uh, only few examples which inhibit the replication process in prokaryotes. Afidacolin, which also inhibits replication in eukaryotes. So these are the action means the specific sites and specific action of these inhibitors by which they prevent the replication process. So this is about the replication of E. coli cell, DNA replication in E. coli cell. Thank you.